Hi, everybody. I'm Michael Carrese, welcoming you to Raise the Line with Osmosis from Elsevier, an ongoing exploration about how to improve health and healthcare. Could studying the DNA of extinct animals, or even bringing them back to life, help us save today's endangered species? Well, that may sound like the premise of a Hollywood movie, but it's work that our guest today, Dr. Beth Shapiro, is engaged in as Chief Science Officer at Colossal Biosciences, which describes itself as the world's first and only de-extinction company. An internationally renowned evolutionary molecular biologist and paleogeneticist, Dr. Shapiro is a pioneer in ancient DNA research, and she's successfully sequenced genomes, like that of the dodo, to study evolution and the impact on humans. She previously served as a professor of ecology and evolutionary biology and lead of the Paleogenomics Lab at the University of California, Santa Cruz. She's currently on leave from those posts to lead Colossal's de-extinction and conservation science teams. Earlier this year, she was elected to the prestigious National Academy of Sciences and has received numerous other honors, including a MacArthur Genius Grant. Well, thank you so much for joining us today, Dr. Shapiro.
Thanks for inviting me.
We always like to start with learning more about our guests, and so we want to know what first got you interested in the study of evolutionary molecular biology and paleogenetics.
It's actually a long and winding road. I started off in broadcast journalism. When I was in high school, I worked for a local TV company. I even got a job as the news director at a local radio station when I went to university. I went to University of Georgia because they have a really top-notch broadcast journalism program. But I had an opportunity after my first year there as a freshman to take a class. It was an honors program class in geology and anthropology. And it was a field program. We started off on the East Coast. We drove across the country, drove up the West Coast, drove back across the country, slept in national parks, and learned about the ecological, evolutionary, and anthropological history of the U.S. And I thought to myself, as I was watching these landscapes and really noting how people had modified these landscapes, not once, not twice, not three times, but multiple times, these are the stories that I want to tell. I want to be a science journalist, not a regular journalist. And since I already know how to be a journalist, I'll just become a scientist. And then I never really stopped doing science. So maybe I'm still waiting for that about face where I can get back to journalism.
Yeah. Your work involves studying a really wide range of organisms, from influenza to mammoths. I would say that's a pretty big range. How do you explain to the uninitiated what you do as a paleogeneticist?
You're right, it's a very big range. And this is part of because of my history. I did my PhD at Oxford University, and I was part of a computational genomics group there that was developing new tools to be able to estimate the rate of evolution with DNA sequences that are sampled at different time points. And what you need is an evolutionary rate that is fast relative to those time points. So if I sequence mammoths that are 100,000 years old and 50,000 years old and 10,000 years old, that time in between those DNA sequences I can use to estimate how fast evolution is happening. That same approach, weirdly, I can use for RNA viruses like influenza and HIV, but sampled over decades instead of tens of thousands of years, because the rate of evolution of viruses is so much faster. I can still get enough information to learn about how evolution is proceeding from 20 years of influenza data or from 20,000 years of mammoth data. So it's the same computational approaches to answer and ask these questions. What I'm trying to do is use the past as a kind of completed evolutionary experiment so that I can make more informed decisions about what might happen in the future. So whether it's about mammoths, how did species respond to really rapid periods of climate change, or the first appearance of people in their habitat? What happened to them evolutionarily? And once I know that, can I use that information to better manage how I spend the money that I have right now to protect species and ecosystems into the future? And for viruses, it's really similar. If I can understand the trajectory of evolution over the course of the last 10 years, then I can use that information to better predict the trajectory of evolution over the next 10 years. And I can use that to make informed decisions about vaccine development or about where I might want to survey birds to look for bird flu that's moving around with migrating flocks of birds, things like that.
So there is a way to apply this knowledge, obviously. I mean, that's kind of your goal, is to use this as a resource with which to make decisions and find the directions you want to go in.
Absolutely. Yes. It's not just about learning about the past. It's about learning about the past so we have more validated scientific information that we can use to predict what we can do to better influence the future.
Right. Fascinating. So just to set the table a little bit here, I think it would be helpful to have you explain a couple of terms I used at the beginning, including de-extinction, which might be new to a lot of folks, and conservation genetics.
Right. Conservation genetics is a little bit easier. This is just the idea that we're going to use DNA information to make more informed decisions about protecting species into the future. I think a common example that people have heard of is the Florida panthers. So Florida panthers in the late 20th century were really suffering the impact of inbreeding depression. So there were very few of them. So they were mating with close relatives just because there wasn't any other choice. And all of these really bad phenotypes, traits were emerging in the population. They had kinked tails, they had weird cowlicks, but they also had things like increased rates of heart disease. They had non-descended testicles that were causing them not to be able to reproduce. And one way to rescue this population is to add additional DNA data. So we can use genetics to understand what it is that's causing these bad phenotypes. We can use genetics to predict whether there are other related populations of individuals that we can introduce to replace these bad DNAs, the bad genes that are in this population. And that's essentially what U.S. Fish and Wildlife did. They took panthers from Texas, they moved them into this population. The females mated with Florida panther males, and a lot of these bad traits disappeared from the population, at least for the short term. So that's an application of genetics and genetic rescue to conservation.
Gotcha.
De-extinction is kind of at the extreme of that. De-extinction is the idea that when species become extinct, you lose really fundamental interactions between species that existed in that ecosystem. So an ecosystem is more than just the species that are in there. It's how they all interact with each other to make that ecosystem robust and resilient to whatever is happening, either from people's perspective or because of changes to the climate or whatever, right? And so when a species goes extinct, some of those roles disappear. So the idea of de-extinction is to bring back those traits. It's a functional idea. So we can functionally replace those missing ecological interactions by bringing back species, by taking a species that's alive today and editing its DNA so that it resembles those extinct species in these functionally important ways so that we can replace those missing ecological interactions. Of course, when people think about de-extinction, they probably think about Jurassic Park.
Yeah, right. I was going to try to get through this without mentioning Jurassic Park, but
I don't mind it. I think it's actually really useful, though, because people understand what we mean by de-extinction when we say Jurassic Park, but it's not actually the way it works. So in Jurassic Park, the scientists were finding dinosaur DNA in the blood of mosquitoes that were preserved in amber. Of course, there's no dinosaur DNA because all dinosaur remains are replaced by rock, right? So there's no DNA there. Also, there's no DNA in insects preserved in amber. I've actually tried this because, what rational
why not?
ancient DNA scientist wouldn't. But yeah, they're too old. DNA doesn't survive well in hot places. And so it's just a really bad environment for DNA preservation. As an aside, the oldest DNA that has been recovered from a bone is from a mammoth bone that's probably between 1 and 2 million years old.
Wow.
And that mammoth bone was preserved in permafrost and frozen dirt since the animal died. And that is really what has made it possible to recover DNA for that long. But that is really pushing the limits. Most of the DNA that we get, even from cold places, dates to within the last 100,000 years. So way too recent for dinosaurs. Right. Anyway, I digress. Jurassic Park, they got the dinosaur DNA. They stitched them together, kind of just like shoving the bits of DNA together. And when there wasn't any DNA there, because it wasn't preserved, they filled it in with frog DNA. Now, that's a weird choice because even then we knew that birds are dinosaurs, right?
Yeah,
not frogs, but
right.
Whatever. That's the idea. You
should have been a script consultant on this movie. I don't know what's the matter with him.
and then it was fine after that. That's not how we're doing it. Instead, for the Mammoth Project, for example, we start with something that is already really mammoth-like. Asian elephants are the closest living relative of mammoth. They shared a common ancestor only around 5 million years ago. That means they already share more than 99.5% of their DNA and their genomes. So what we do is we go out, we collect mammoth bones, we extract DNA from those, and we sequence a whole lot of really tiny broken fragments of old DNA, and then we align them to a reference genome of an Asian elephant. And then we can ask, using a computer, where are all the mammoths the same as each other, but different from Asian elephants? So these are probably DNA sequence changes that are important for making mammoths mammoths rather than elephants. And then we use the tools of genome editing to take a mammoth cell that's growing in a dish in a lab and gradually cut and paste our way from the genome sequence of an Asian elephant to the genome sequence of a mammoth at the places where it's really important to bring back those key characteristics that defined mammoths. So for the case of an Asian elephant, making it able to live in colder places, having this characteristic furry shape. The shape of the tusks was a little bit different. The shape of the head was slightly different, but mostly focusing on adaptations that would have allowed that animal to live in cooler climates, because that's what we want, is an elephant-like animal that's capable of surviving in cooler climates to restore those missing ecological interactions that a mammoth once had with other members of its ecosystem.
So if this is successful, what's the vision? To have, mammoths roaming northern climes again?
Yes, that is the vision, and it is a long-term vision and goal, of course, because mammoths and elephants are very slow growing and slow. They have a 22-month gestation and they reach sexual maturity as teenagers. So in order to have a community, a population of animals that is sex structured and age structured the way elephants will be, this is a many decades in the future sort of project. But yes, with all of the projects that we've launched at Colossal, the goal is to have populations of animals that are able to thrive in ecosystems of today in the habitats where they once lived.
So I don't want to be flippant here, but I'm sure people listened to your description of what you were doing, stitching together mammoth DNA and all this other kind of stuff, and what could go wrong, right? So what could go wrong with what you're talking about?
Well, I think the slow pace with which we're doing this is trying to mitigate anything that could potentially go wrong. I mean, it's a very odd way of asking the question. There's obviously risks associated with editing DNA because we're changing DNA sequences on a different genetic background. But there are a lot of tools we have at our disposal to be able to evaluate and potentially mitigate those risks before we make those edits. A great example is our dire wolves, Romulus, Remus, and Carrese, who we announced in April this year. So we learned by sequencing the DNA from fossil dire wolves that they had a light-colored coat. And we wanted, therefore, to make Romulus, Remus, and Carrese have light-colored coats. But the gene in which fossil dire wolves had this variant that tells us that they're light-colored. If we look at living gray wolves, and it was gray wolves that we used as the genomic surrogate for our dire wolves, they have an instance where there are particular variants that appear in those genes. It's not the same variant as we saw in dire wolves, but it's in the same gene. So we thought it was a little bit too risky to do this. The gray wolves can be blind or deaf. And so we made the decision not to make the dire wolf specific variant to recreate this phenotype, but instead to look into variants that already exist in healthy gray wolves that make them have light colored coats. And that is the path we used to de-extinct this light colored coat phenotype. So we care very much about, obviously, we care about animal welfare. We wouldn't be a very good company if we were making animals that were sick. So fundamentally, we want to make sure that as we are bringing these species back, bringing back these key phenotypes, we're doing it in a way that leads to healthy animals that are able to thrive today.
Yeah, do no harm sort of approach. So you mentioned the company. Tell us about Colossal Biosciences and the mission, and beyond what you've mentioned already, your role specifically there.
Colossal was founded almost 4 years ago. I joined a little bit after it founded as a consultant, a sort of lead paleontologist consultant. I think, again, because I wrote the book called How to Clone a Mammoth, and they initially launched to clone a mammoth. I joined full-time as the chief science officer about a year and a half ago now. Colossal is a de-extinction company, but it's a species preservation company with the goal to make extinction a thing of the past. It's a laudable, very big goal. What it means, though, is that every species that is a candidate for de-extinction has a different set of technical and ethical and ecological challenges that will need to be solved in order to get there. What I like about the projects that we've launched, we have the mammoth, the thylacine, which is also the Tasmanian tiger, and dodo and moa. Those are two avian projects. Those were more recently launched. And because each of these requires a different set of tools that have yet to be developed, as we develop them, we now have new tools that we can use to genetically engineer for the purposes of conservation pretty much the entire animal tree of life, with the goal of helping species not become extinct. A great example of this is last week, we submitted a paper and put on the bioarchive, the public repository, a new technology that our avian team developed to be able to culture primordial germ cells. These are cells that will eventually become either sperm or eggs, depending on if it's a male or female, in a dish for pigeons. This is really important for the purposes of de-extinction and genetic engineering for avian conservation because we can't clone birds the way we clone mammals, like Dolly the sheep. So with the mammoth or with the thylacine, we can take a cell, any type of cell from anywhere in the body, make the edits that we want to it, and then clone that cell using somatic cell nuclear transfer, that process that most famously brought us Dolly the sheep. We can't do that for birds because we don't have access to an egg cell at the point where we can remove the nucleus of that egg cell, the first step in somatic cell nuclear transfer, and then transfer the edited nucleus there. We just don't have access to it because of the weirdness of bird reproductive biology. So the way that we can edit birds is we have to edit the cells that will eventually be sperm and the cells that will eventually be eggs separately, and then put those into a bird and then breed those birds together, and they will lay an egg that eventually has our edited dodo. Or if it's a conservation project, for example, our paired conservation project with the dodo project is the Mauritian pink pigeon, which is a highly endangered pigeon species that also lives in Mauritius. The dodo was a type of pigeon, in case you're wondering why we're focused on pigeons.
I didn't know that.
So we now have the technology to do that. What's weird about this is we knew how to do this for chickens. Mike McGrew's group at Roslin Institute developed the recipe for keeping chicken PGCs in culture 20 years ago. And then we had no other ability to do this for other birds until relatively recently, when Mike's group published a recipe for geese, and now our team has a recipe for pigeons. It's super weird that the recipe doesn't work for all birds, but it doesn't. Mm-hmm. But now that we have a pipeline for coming up with the recipe, and it's just a different combination of metabolites and transcription factors and other things that go into this culture to keep the cells happy, now we have this incredible breakthrough that will allow genetic engineering technologies to be applied to birds. And birds are among the most endangered species on the planet. So it's about time that we start having the type of biotechnological resources that we need to help them to be able to fight extinction.
This is just, it's like mind-bending. I can think of 5 other cliches for it, but it's just absolutely amazing what you're doing. So one thing I'm curious about is that these ecosystems have adapted, right, to the absence of all these extinct species. And that's been going on for a very long time. So what is the thinking about what happens when these species are reintroduced? What are the possible upsides, downsides, and what have you seen already along these lines?
It's a good question. And it's absolutely true that ecosystems are always in a state of flux and they're always changing. And we as people introduce species to landscapes, sometimes on purpose and sometimes by accident. And we've been doing that for a long time. I think one of the important things to think about, though, is that when these species disappear, there is a cascading long-term impact on other species that are there that we can help to manage if we reintroduce these species. Take, for example, what the Mauritian Wildlife Foundation, who is one of our conservation partners in Mauritius, are doing with some of the landscapes that we are thinking of eventually having dodos to reintroduce. So at the same time as dodos became extinct, they lost the Mauritian giant tortoise. And this is because when people first arrived in Mauritius, we brought with us things like rats and cats and pigs, some on purpose, some by accident. And dodos and giant tortoises laid their eggs in nests on the ground. And these made really easy meals for things like rats. And so in order to reintroduce giant tortoises to these landscapes, they've had to do a ton of non-native species removal. So coming up with ways to remove rats from this landscape, to keep domestic cats and macaques out of these landscapes because they all consume the eggs of these native species. And they haven't de-extincted giant tortoises, but instead they moved giant tortoises from Seychelles, a really closely related species that plays the same sort of ecological roles as the extinct Mauritian giant tortoise would have. And what they've seen is that when these giant tortoises are back on the landscape, the work that they are doing to remove non-native plant species by hand gets this incredible boost from the tortoises being present. And this is because the introduced plant species didn't evolve alongside giant tortoises, so they don't have any evolved mechanisms of not being eaten by them while they're babies. And so the tortoises are just eating the crap out of all of the non-native plant species.
Oh, wow.
But the native plant species that did evolve alongside giant tortoises do have ways of avoiding being eaten by these giant tortoises. They have little serrated edges on their leaves that make them unpalatable. Maybe they sequester toxin in their leaves. And so what we've seen in these habitats is that the native plant species have started to come back. Even the ebony seeds, we now know they will germinate more readily after they pass through the digestive system of a giant tortoise. And so the ebony forests are coming back. And so, yes, there has been some flux that's been caused by the reintroduction of giant tortoises, but this flux has really benefited the species in the ecosystem that were there in the first place. And the cost has been the species that people have been trying to remove to get to reestablish the habitat the native species need.
Amazing. So what is your work telling us about the risk of zoonotic diseases? And I bring that up, of course, because, that's still one of the active theories, leading theories about the origins of COVID-19.
Well, I think, zoonotic diseases are becoming more prevalent as people push into habitats where they weren't before. So as ecosystems become more destructive, we see zoonotic diseases emerge in sites where deforestation is prevalent or where there is much more interaction between wildlife and people. And I think reestablishing healthy ecosystems and reestablishing at least some barrier in these ecosystems between people coming in and taking things out and the wildlife themselves will be there. One really interesting thing about the ability to use genetic engineering for wildlife as well, it kind of addresses this question, but it's a slightly off topic, is we've also introduced diseases that are impacting wildlife. So, for example, in Hawaii, the honey creepers are in danger of becoming extinct because of the introduction of avian malaria. There weren't mosquitoes there. They didn't have avian malaria until people brought the mosquitoes and the avian malaria there. But our advances in technologies that will allow us to edit the genome sequences of birds will make it possible to help those bird species become resistant to avian malaria if we can edit their DNA to provide genetic resistance. The same is true for work that is being done in the US with black-footed ferrets. Black-footed ferrets are one of the original, they call them the class of 1966, the first species to be listed in the Endangered Species Act. And they are becoming extinct because of reduction in population size, but also because of plague. So plague was reintroduced into their habitat or introduced into their habitat as Europeans expanded to the west. And plague is carried by prairie dogs. That's the favorite meal of black-footed ferrets. But domestic ferrets are genetically immune to plague, presumably because they evolved alongside it in Europe. And this is the evolutionary cousin of the black-footed ferret. And so there's work being done right now by the nonprofit Revive and Restore, the Smithsonian, Fish and Wildlife, San Diego Zoo, really great team. This is not a project that I'm involved with personally, but I've been tracking it and I really love it, to identify what it is genetically about the domestic ferret that makes it not able to get plague, so that we can use these same tools of genome engineering to modify the DNA sequence of black-footed ferrets so that they can live in the habitats of today that include plague, but not get plague, or at least not die from plague if they were to get it. But I think it was really exciting work both in disease and in ecosystem restoration that comes out together. And of course, the tools that we're developing, all of the tools that we need to make hundreds or thousands or even tens of thousands of changes to DNA sequences at once, the computational tools to better understand the risks of the different types of modifications that we're making. These obviously have immediate application to using gene editing for human medicine and for animal medicine.
Yeah, right. Right. That's a very, very good point. You are an author, and one of the books you've written is called Life as We Made It. I think that's your most recent book, and that explores genetic modification in the context of history, archaeology, genomics, what we've been talking about. So with that book particularly, what are the things that you're hoping to get across to readers?
I think with that book, what I'm really thinking of, there's a lot that we hear about the risks of using gene editing and genetic engineering, both for conservation and for medicine and for agriculture. And one of the common things that we hear is that we shouldn't be messing with nature. We have a responsibility to stand back and just let nature do its own thing. And I like to make 2 points with that book. And the first is that we have been messing with nature for as long as we have existed as a lineage. We took gray wolves and turned them into Chihuahuas and Great Danes, and we took corn and turned it into teosinte, and opposite way around there. But many of the things, everything that we've done has really been to make the planet a better place for people. And if we want a future that has a rich biodiversity of life that isn't human, as well as the capacity to support 10 billion or more people, we have to be able to transform less land into more food for ourselves, for our animals. We have to be able to figure out how we can help species adapt to rapid changes to their habitat, changes that are happening faster than evolution can keep up. It's too late for us to step back and allow nature to take the time and space that it wants to get better, because our footprint is too big. But it's also not different from what we and our ancestors have been doing for tens of thousands of years. I mean, it is different in a way because we're fundamentally altering the DNA sequence in a very precise way, rather than the imprecise way that we've been doing forever, which is just by selectively breeding the things that we like together, right? So there are differences. It's also important to remember when we think about the risks of adopting new biotechnologies for the purposes of agriculture and conservation, human medicine, that there is also a risk to making the decision not to use these technologies, right? I think too often we think one of these things is risky and the other one is safe. The other one, where we just let things proceed as they're happening right now, is decidedly not safe. We know the future if we keep doing exactly what we're doing right now, and that is a future with high rates of extinction, high rates of illness among people and among the animals that we interact with and plants that we interact with, and a reduction of arable land that will lead to starvation in different places in the world. I think that is riskier than allowing ourselves to do whatever we can to fully understand the potential of the technologies that are at our fingertips.
Well, you've persuaded me, if that means anything. We're running out of time, so before we wrap up, we want to give you the opportunity, we give all our guests, to provide a little advice to our audience, learners and early career professionals about their careers and thinking about taking opportunities. And I mean, you're a perfect example of somebody who was on one track and got interested in something else. And you're doing stuff now I'm sure you did not imagine was possible, 20 years ago or whatever. Yeah. So,
or even 5 years ago, right?
So young people ask you about building a career. What do you say?
I think, it's a tough time out there for science, for people like us, people who are in the medical field or people who are in ecological fields or people who are just thinking about basic science, to imagine what the future is going to be like 10 years, 20 years, 30 years down the future. I think it's important to keep an optimistic outlook. Remember why you're doing this, and remember that you can do this work within your value system and that what you're doing will make a difference. And I just think it's important right now, especially, to not get bogged down by all the negativity that's out there. When I think about what Colossal is doing, one of the biggest benefits that I've seen is that People who are in middle school and high school and college students who've only been told that the future is bleak, that extinctions are happening all the time and there's nothing they can do about it, they really respond to Colossal's work because it takes control back. It gives people a sense of optimism. It tells people that there are things that we can do to make sure that the future is better. And anybody who is a scientist right now needs to hear that. So wake up in the morning and tell yourself that there is something that you can do. The future is brighter. There are all of these super cool, exciting tools that are at our fingertips that you can be part of developing the tool, developing the regulatory framework in which we can work with those tools and making sure that they are applied equitably and fairly and within your value system to help make the future a better place.
Well, it's a wonderful bit of wisdom to leave our audience with. And I can't thank you enough for your time. I said before we started recording, you've probably got the coolest job of any of the 500 people we've interviewed.
I think it's pretty cool.
We'll definitely be keeping an eye on it. But for now, thank you so much for joining us today.
Thank you.
I'm Michael Carrese. Thanks for checking out today's show. And remember to do your part to raise the line and strengthen the healthcare system. We're all in this together.
If you like this podcast, please share it on your social channels. You can also subscribe to the series and check out all of our episodes at osmosis.org/raisethelinepodcast.